Hi everyone, my name is Koay Kian Yew, metric number A165404. Today I am going to present video seminar with title Whole Genome Sequencing of Mycobacterium Tuberculosis Isolate and Clinical Outcome of Patient Treated for Multi-Drug Resistant Tuberculosis in Tanzania. For introduction part, tuberculosis, particularly multi-drug resistant tuberculosis or extensive drug resistant tuberculosis, is still a global medical emergency. There are some PCR probe-based assays can be used, however they have limited anti-TB drug susceptibility value, especially if the mutation occurs outside the target region. And this is why whole genome sequencing comes in as an alternative method for diagnosis. There are benefits of using whole genome sequencing. For example, it does not affect the anti-TB drug susceptibility value. Next, it can also identify genotype predictive of drug resistant phenotype within the entire region of microbial genome. Whole genome sequencing has also potential to determine genetic relatedness and identify transmission dynamic necessary in guiding clinical decision. For the methodology part, the study setting is shown in the table. Study design, population, and recruitment of participants. In this study, we were able to get total 87 total participants. 57 of them are resistant to anti-TB drug, while 30 of them are susceptible to anti-TB drug. In this data collection, we are through standardized semi-structured questionnaire. Pre-treatment data was collected, while the laboratory procedure involved sample collection, culture of STBC, and phenotypic drug susceptibility testing. 200 microliters of sputum sediment were inoculated on two slopes of LJ medium containing either pyruvate or glycerol. For the DNA extraction and whole genome sequencing parts, we are using a modified protocol based on restriction fragment length polymorphism (RFLP). For the bioinformatics and genomic data analysis, quality control and mapping of reads were characterized using Kraken version 1.1. Variant calling and in silico resistant and lineage typing was using Lofric version 2.1.2. Phylogenetic reconstruction using SAMS2 BCF2 version 1.9, ExamML version 3.0.21. For the analysis of clinical data, Stata version 14 is used. We are now focused on results. Based on the table 1, we know that the total 87 of participants, 40 are male, 47 are female. 20 of them co-infected with HIV has the median CD4 count, which is 246. Based on table 2, we know that there is association between the previous history of TB treatment and phenotypic drug resistance. Based on table 3, we know that 57 of them have at least one mutation in the gene for the 7 anti-drugs. The anti-drugs is shown in the red color, while the targeted genes are shown in the blue color. For the table 4, we know that 47 of them received a successful treatment, while 7 of them as defaulted. Unluckily, 3 of them died after 9 months of enrollment. 2 of them are from multi-drug resistant tuberculosis, while 1 with polyresistant. From the figure 1, we know that the predominant is lineage 3, followed by lineage 4, lineage 1, and lineage 2. In table 5, we know that the spolygotype from lineage 3 is called as Central Asian strain, lineage 4 is called as Latin American Mediterranean, lineage 2 is called as Beijing, while lineage 1 is from East Africa, India. There are some additional discussion. In this finding, we found that high concordance between whole genome sequencing and conventional culture-based DST in predicting phenotypic drug resistance to anti-TB drugs. There are also higher success rate, which is 30% compared to the global multi-drug resistant TB treatment. In this study, we also found that 29.9% of the study participants have low BMI and 23 of them was infected with HIV, but they have shown no effect on multi-drug resistant TB treatment outcome. In conclusion, this study offers the comparison of mutation detected by expert and whole genome sequencing with phenotypic DST of Mycobacterium tuberculosis isolate in Tanzania. As expectedly, whole genome sequencing was robust in ruling out the drug-resistant tuberculosis, especially in the absence of detectable mutation. At last, whole genome sequencing provides rapid and comprehensive diagnosis of drug-resistant tuberculosis. That's all for today.